Oh, well, hello. I'm Allison Nowak, and I'm here to train you on one of the most fun academies, one I have been teaching for a while and truly, truly love, called Forensic Science, also known as the CSI Experience. Okay, so Forensic Stream Academy, so much fun. What can we expect to encounter? What can you find in your kit? What are your students going to get to explore and analyze and figure out in the case studies? So much to do, so much fun for both teacher and student. So, with that being said, let's get started. What can you expect to find within your kit? It's a pretty hefty kit. You have a lot of things in there. And of course, listed in the appendix is the full list of what it is that you should have in your kit. There are some things that are not listed on there, which are very helpful for the different experiments, such as the unknown powders experiment and others as well. So lots of little things that you can utilize for any of the experiments, and also to teach your students how to make certain concoctions that will help them to figure out what a liquid is or a powder is. So much fun. So here's just a basic list. Like I said, you can find this in the appendix of your binder, which will be included in your kit. Just to show you what the binder looks like, here it is. And you have, like I said, in the back an appendix just after your case studies that will tell you the materials that are included in your kit. Now, that being said, here's all of what I've listed. You have some rubbing alcohol, tweezers, snack-sized Ziploc bags. You have Petri dishes, a couple of these. Some foam trays that you can set up all of the different things that your students are going to need to accomplish the tasks at hand. You will have a very nice, fresh, brand new, so be careful when doing fingerprinting, a brand new, nice ink pad. You will have pipettes, come in very handy for many things, as well as you'll find things such as your bottle of white vinegar, a bottle of lemon juice. You'll see there's a bottle of ammonia, as well as iodine, so you can create your iodine solution when you're working on the unknown liquids experiment. Also, you have things such as baking soda, you have baking powder, you have cake flour, regular flour, salt, gloves that are, of course, latex-free, rubber bands, cheesecloth, vegetable oil to get that ink off of the hands of your students. Comes in handy, works for some people, others maybe not. It might take a few hand washes after also using the oil to get that ink off of your fingers, but the kids always enjoy the fingerprinting and take pictures of themselves, with the ink on their fingers. So it's really just fun anyway, even if it doesn't come off right away. Then you'll also find in there some manila folders to help you to organize all of your case studies. I personally use them to do the case study, and I hand it out almost like it's a case file that was being handed to that CSI tech, for them to investigate and go through the different evidence. I'll show you more about the case studies later in this video. You will also find fingerprinting cards and other things that you may not find. Oh, Dixie cups, very handy as well. And magnifying glasses, which are great for when you're doing the fingerprinting. Things that you might not find in the kit that you can find in your science lab would be things such as test tubes and beakers, things like that. So you want to make sure that if you do have a science lab or if the science teacher has these items available, that you get those situated before you do the experiments that require them. Okay, so now you're into the binder. The first week is all about evidence gathering and observation. Observation is absolutely critical when it comes to making sure that that data is properly collected, the information is properly collected. So there are a couple of fun little moments within this first week where you can have your students observing and doing almost like a game, who can see the most with a couple different scenes, as well as you're going to be going into and exploring what it is to truly be a CSI tech or a CSI agent. What do they wear? How do they have to act on a crime scene? And this brings into question what students already know. So that's why I like to start with, what are the crime scene investigation basics? What do students already know? I'm all about the prior knowledge. I like to see what they've seen or hear what they've seen. What shows do they watch? Do they watch CSI? Do they watch Law and Order? Do they watch Bones? Do they watch all those different things that are out there? This day and age, there are so many different crime scene and police work type of shows that I'm sure that you're going to have more than one student saying that they've seen at least one of these shows or heard of it. And so with that being said, I like to kind of give them a brief history of what is forensic science, where did it come from, what are the different fields, and then I really like to look at a compare and contrast kind of situation, TV versus reality. What is it that you've seen on TV? Now that we've gone through this, what they actually do via the YouTube links that are available inside of your binder, once they've gone through that and they see what it's really like or what it is that they really wear, that sort of thing, then they can compare and contrast, okay, what is it that I saw and how is it really? Very important to make that distinction. So with that being said, you have a compare and contrast example. So they talk about, bring to the table, what have they seen, what do they know, or what do they think that they know. Watch the YouTube video, seeing what it actually is, and then play a fun Kahoot game, which tests what they just saw. So that is a fun thing. All of the links are in your binder in week 1 under evidence gathering. Then you go into some observation. There are 2 image studies. One is an office scene where the person has been murdered, and the other one is a doctor's office, which is just very strangely laid out. And you'll see this within pages 3 and 4 in your binder in the academy itself. What comes along with that is the what do you notice questionnaires, which is, for example, what color was the coffee mug that you saw in this scene? How many people were wearing shoes in this scene? So some really funny little questions, but it's really bringing their attention to the details. And it's all about details and accurate information gathering. So you're already putting that into their minds, and they are having a little bit of fun to see who noticed the most in those 2 scenes. That being said, I would like to stop there just to make sure that I show you something extra that I like to do during this week 1. Like I said, what is the history behind forensic science and what are the different fields within it? Because a lot of the time, your students don't know that forensic science is just a big general umbrella field, essentially, because there are so many different ones within this field. I like to teach them about where those came from, who contributed to them, and what all these different fields are. Okay, so this is my own personal PowerPoint that I like to use. It is specifically from, it's copyrighted, of course, 2017, Aaron Mucci, but it's a really great, thorough presentation to, again, touch upon all the different disciplines within this field of forensic science, as well as to teach the students about where it all started, who contributed to those disciplines, and who overall contributed to the field and what they do today. So I go through this just briefly. I don't really take a lot of time. It maybe takes you 10 minutes to go through some of the things that you may want to, but it's fun because students don't really know all of the ins and outs of this. So the dictionary version, of course, then you have more of a definition or more of an explanation, as well as a synonym, which is for forensic science. You can also call it criminalistics because of that connection between civil laws, criminal laws, and science. Then I go through what is the role of science within this, and the idea, again, of being accurate and objective and making sure that information that they collect allows the investigators to piece together events accurately and without a doubt. Then I go into, here are the different disciplines, some of which the students are very familiar with or are aware of, whereas others they might not have heard of. So they might not have known that, sketches and recordings, that sort of thing, that art was a big deal within this. Firearms, they usually know. They don't know what the word serology is right away, but they do know that blood has to do with forensics, of course. Impressions is always a good one because they're like, okay, what is that? And I have to explain it's, you can be identified by your teeth or bite marks or things like that. Fingerprinting, they always come up with that one. They don't always know about forgery and question documents, so that's an interesting one for them to learn a little bit about. And then I really like to focus in on trace evidence because that is one thing you will continually touch upon and go back to as you're doing fingerprinting, etc. So just knowing that any little piece can be left behind. Hair, fibers, soil, plants, and that's really a big deal. Trace evidence is huge within this field. So then I go into the history. And this part is just, like I said, I go through this very quickly, give them an idea of the contributors, who contributed to each of the fields. And, it's not that you have to necessarily test them on it, but another good thing you could do if you're using this or if you're teaching them these different backgrounds and where things came from, you could always do another Kahoot. Create a Kahoot yourself or put one together, maybe do a Quizlet or something like that to make it fun where they are kind of going back over and trying to basically reproduce that information somewhat. But again, it's not something to stress upon and it's not something you have to do. This is just something extra I like to do and the kids always appreciate it because they just get even a better sense of what this field is and how much there is in it. The one that you do want to stop on is, of course, Edmund Locard, and that goes back to the trace evidence idea. So with Edmund Locard, you tell them about him, hear from this, and you go into Locard's exchange principle. Explain what it is, and then, of course, there's a YouTube video attached there about transfer of material and trace evidence. This specifically is the one you can find in your binder. And then, of course, this is the Kahoot that was provided as well with your academy. Again, it's up to you which one to use. If you take a look at this Kahoot and you find that you want to put more to it, then you are more than welcome to create your own. Like I said, I like to create my own, but I do start with this one to see what they got from this video that I play. However, there's a lot in the video that I felt, you could add, so I ended up making my own as well. So I recommend that to you. If you're a big fan of Kahoot or Quizlet and you like to use those games, then I would recommend making one so that it fits whatever it is that you want your students to get out of what you're showing them here with the history and the contributors of the field. So there's a lot more that's in the PowerPoint that I have personally downloaded and used. I'd like to kind of go into some things such as, how are crime labs organized? There's different types of crime labs. And then I touch upon the FBI, the Federal Bureau of Investigation, and who created it and why it all started. Because, of course, it has to do with the forensics and data gathering. So it does connect very well just to touch upon these things. Again, all of those that I just showed you, those slides in that presentation, it's something quick to go through. It's not something that you need to dwell upon, but it's really good information for the students to just get an understanding of where everything within this field, where each of the different disciplines have come from and how they may be used in, of course, data gathering today. Week number 2 is all about TOD. There is an equation that is given to you, formula, that the students can use to practice figuring out based on information that they're given about the person and the events and the times that things might have occurred. Then they are able to use that, plug it into the formula, and go ahead and calculate out to the best that they can, a very approximate idea of when that person may have passed. It's very fun to do. The students really enjoy it, and I still have a student to this day who says she is the TOD expert, and so it is something that they do enjoy, especially if they're more math-driven students. They're really going to love the calculating time of death segment. And with that being said, you have a worksheet practice as well as you have 3 different cases that the students can take a look at as well. And all of that is, of course, included in your binder. You also are going to go through and explain and discuss what it is to think of a time of death. You'll talk about things such as air temperature, wind, clothes, things that you see. How does the body look? How does the body cool down? You'll learn about rigor mortis with them, or talk to them about that. You'll talk about livor mortis and how all these things play into calculating a person's time of death when they are discovered on the scene. After that, you pose some questions to them, all of which takes about 30 minutes. So that's why the very last part that you're going to be doing takes about 2 minutes, and then 3 to 5 minutes to talk about once they've calculated with their time of death worksheet practice. Again, all of which is outlined and included in your binder. This is particularly page 8, where they will have the formula and the example, and then be able to go from there with calculating their own on the practice sheet. So if I go back over to my own personal one that I like to use, this is calculating time of death, and there is a link to discuss how it is to, or how it was to be a CSI agent. If you go to the first link, which is forensics.rice.edu/index.html, it's a really fun website. It's all based off of the actual show CSI, and it has many different ways that the students can explore the disciplines within forensic science. This particularly for calculating time of death, there is a specific one in the first training part of it where you're going to, as the student, will see and explore, they get to learn how to do time of death with one of the CSI agents from the show. It's a really fun resource, and you can use that over and over again throughout this time if you have downtime, or if you've completed an experiment and you have an extra week or you have extra time. Students can pop onto this forensics.rice.edu and have some fun with exploring the different disciplines. Ballistics, everything is on there. So really, really cool site. I strongly recommend that you check it out, and you'll enjoy it yourself, as well as your students are going to love it. Okay, that being said, I found personally this YouTube video, and it goes through fingerprinting. So I will go back to that in just a moment, because first I want to talk about, of course, Todd. So here's what I show the students, or I have up on the board, so that they can see as we talk about it and we go through it. We go back over converting decimals and making those count for minutes. So the students have to kind of go through this just to remember and understand that it's every, quarter of an hour. So I always show them that. And then I go into, this is right out of your binder. You can find it in the online version, or the drive that you will be included on. And this has everything on there for you. It's a Google document all in itself. Okay, so again, I like to put this up after we go over converting the decimals into minutes. I like to give the kids this so that they can look at that time of death practice worksheet and try to apply this. Now, fingerprints. Here's the YouTube that I had before. Fingerprinting is week number 3, and the students really have a lot of fun with this. This is just a great start to the different experiments that they're going to encounter as they go through this academy with you. So it's really fun that you start here, you teach them a little bit about all the different things that they might be looking for, or you can always have them do the fingerprinting first and then go back to this and use it to go over even more in depth. What is it that you're seeing under that microscope? What is this ridge? What is that? Do you have a delta? Do you have a whorl? Do you have a loop? Do you have arches? Things like that. So you get to really examine them closer, and this helps because this particular YouTube goes through those different points, and they highlight and show you, this is what this looks like, here's one of these, and they pinpoint all those different points that you really want your students to learn about. So what can they expect during the fingerprinting week number 3 segment? What you're going to do during this session is you're going to be teaching the students how to roll a fingerprint as well as take a flat impression. They'll also be able to lift prints off of things, different items if you want to practice it or try it that way. Another way that you can practice it is with coloring. Okay, so with the fingerprinting, lifting prints, this is what students really enjoy doing. There is a way to utilize the pencils that you will find in your kit. You'll find several pencils that you will have the students use to create this. So what they do is they are going to, let me just make myself a little bigger here. They are going to use the pencil and color a nice big rectangle, and really, you want them to make sure that they make it very dark. Then they'll take their finger, roll it or rub it in that rectangle, and then with a piece of tape, they are going to lift the print off of their finger and put that tape back onto the piece of paper where they were coloring. And it'll turn out something like this. Don't know if you can see it, but it'll turn out like that. So it goes from them just rubbing it, lifting it off of their own finger, and then putting that tape on a piece of paper so they can examine it further. For the rolling fingerprints, as well as your flat impressions, included in your kit or in your binder is a wonderful setup for taking fingerprints. So this I would recommend you print on cardstock, just because it'll just be better with the ink, and honestly that's how I've done it each time, and it's really been most successful with putting it on cardstock. So the students learn how to do the different impressions. All of the top ones, of course, are rolled, and down here is where they have the flat impressions. So going through and just really showing them that technique of rolling from one side to the other. Okay, and now if they have too much ink on their finger, you want to make sure that you're telling them not to press too hard, because if they press too hard with that much ink on their finger, they're not going to get a very successful fingerprint that they can then use their magnifying glass and examine. So make sure that they're being light, and especially with those impressions too. Make sure the students also know it's important that they really do get not only the fingerprint, but the flat impressions have to be a little bit further down to the knuckle of the finger. So it's always fun to watch them try to do this, and I like to do it in a way where they're helping each other, teaching each other, and working together. I've even taken it as far as to print out ones that said, you know, it said wanted for so-and-so, their name, wanted for, and then they get to make up different crimes that they may have committed, and they really have a lot of fun with that, just being silly and creating a made-up crime that their friend had committed and then taking their fingerprints as the police officer. So definitely you can make it really fun in your own way. And then you go into, after all of this, you want to make sure that they understand, as they're examining those fingerprints with their magnifying glasses, you want them to understand the different points that make a fingerprint unique. And that is why I like to use that YouTube video, as I mentioned in the last slide. The YouTube video really shows all those different points and different types of fingerprint ridges that the students can then go ahead and see what it is that they have in front of them. So does my fingerprint have a whorl or a loop? Does my fingerprint have a bifurcation? Can I tell where there's a short break? Can I tell where the deltas are? And so forth. Again, the YouTube video is extremely helpful, very informational, and shows the students how to find those unique points that make their fingerprint. Now, at the end of week 3, at the end of your fingerprinting, you'll notice that there is in big red letters, for next week, you will need to purchase strawberries, the largest ones that you can find, and make sure that you put that alcohol, the 91% isopropyl alcohol, into the freezer. The reason that I say that, you have to get the appropriate temperature, and it does make a difference when you're doing DNA week with the strawberries. Your formula that you create is not going to work to extract that DNA if you do not have freezer chilled isopropyl rubbing alcohol. I'm telling you from experience, you have to chill it. It's not something you can just do quickly the day you get into school. You have to make sure that you kind of prepare the night before. So again, make sure you find your strawberries as well as get that rubbing alcohol in the freezer as soon as possible before you begin. Okay, so that brings us to, that being said, of course, with the strawberries, that brings us into DNA week. DNA week is a big one. It's super fun. The students love figuring out how to extract the DNA from the strawberry, as well as you can expand upon it and have them actually extract their own DNA doing a cheek swab and then looking under a microscope at both. It's really an exciting, very cool experiment, well, not experiment, but activity that they really get to enjoy. So, of course, last session is about fingerprinting. So remember that the students did their fingerprints with each other and maybe made up fake crimes, etc. With those fingerprints, what you can do to begin your session for DNA week is you kind of want to go back to, okay, what did we learn last time? So with the fingerprints, around the room you can put up the different fingerprints from each of the students, and you will give them about 10 minutes to try and match the fingerprints to those classmates that the fingerprint cards are in front of the room. So you kind of have an answer on a piece of scrap paper for yourself so you know whose are whose. And but see how many the students can figure out just judging by what it is that they have. Again, that's about 12 minutes. You'll see the explanation for that on page 11 of your binder to start. So again, something fun to do. They did all those fingerprints last time, so why not utilize them and have the students have a really great time trying to guess whose fingerprints are whose. Then you get to extract the DNA from a strawberry. So how do you do that? Let me explain. So what is this process? And something important that students need to understand is that it is a very intricate process to extract that DNA, and there are reasons why this is extremely important for scientists. So you pose these questions to the students. Okay, what is the process? Can you do this process? Can you, of course, implement the process and accurately? And then why is it that this is important for scientists? Then you can discuss and observe the differences in the DNA strands. I like to show them different DNA from all different species and just talk about, what are these differences? What are the differences? What can you notice? So that's something, again, you can do on your own, add on to what you're talking about within this particular week and session. Outlined on page 12 of your binder is exactly how to extract the DNA from a strawberry. And this takes about 15 minutes to do. If you follow the students, if you give the students each a list, which is also included for them, there's an extra on just after page 12, there is an extra list for you to go ahead and copy and give to your students so that they can either follow it on their own, or you can be dictating and leading them through it. It's however you feel you'd like to go ahead with it. You can either, like I said, make it self-guided or a teamwork type of thing, or you can go ahead and guide them through it and do it all of you together. My students have a lot of fun once they've already extracted the DNA and they've gone through the process. They really love to name their DNAs in the Petri dishes, just something silly that they've always done, and just gives them, again, something a little bit extra to go along with it. Anyway, so that being said, we have then in your binder another example, sample lesson, I should say, this time, about how to extract your own DNA. So then after you've done the strawberry, students can take it a step further and learn how to take DNA from themselves or a human in general. And there's lots of extensions and reflections that go along with the DNA week. So I strongly recommend that if you do have the extra time and you can do these extensions and expansions, they're really fun and the students really do love it. Sometimes they already have that prior knowledge from science class. They've been working with DNA with the science teacher. So that's always fun for them to be like, oh yes, I remember this. And then they're just continuing to connect. Now, at the end of DNA week, you'll notice yet again on page 15 in big red letters that you will need to make the universal indicator before the next week. You will have to go shopping to get the cabbage. That means you will have to go and find, you can buy it very easily at Tops, Wegmans, any of your local grocery stores, some red cabbage. Red cabbage is ideal because the universal indicator that you're going to create is a dark purple, so you need the red cabbage to get that dark purple color. Okay, so now we're in the really fun experiment phase. Week number, I think we're on number 4, is your Mystery Solutions lab. So Mystery Solutions is a very fun but very quick type of experiment lab for the students to do. They really do have a lot of fun testing out and seeing the different liquid color changes and so forth. However, I do allow them to do a little bit of mixing at the very end because it goes so quickly. So they love to see the different colors and reactions that happen from doing that also. Anyway, though, I want to stick to the solutions lab itself. So the first thing, like I mentioned, is you want to make sure that you purchase red cabbage to make your universal indicator. To make the universal indicator, you will find the instructions in your binder. It doesn't have a page number specifically on it, but it says Unknown Solution Lab Instructions. It's basically the lab, which is starts just after your Unknown Liquids lab activity with response chart. Those cards that you will give out to your students. So it says, of course, you want to make your universal indicator first before that class. You also want to make sure that you have read through the entire Mystery Solutions lab, as well as that you are prepared and have printed your cards for the students so that they will have a copy of how these solutions react. That would be these ones. I like to print them and cut them and just have them placed on the table for whichever student, if they're working together, or if they're doing it individually, just making sure that I have at least 2 on every table or a couple per, group of desks so that they know what the reaction means when they mix the indicator and the solution in the test tube. So again, this is what it looks like. You'll find that inside of your binder. Also important to have is the lab process itself, so the whole entire activity and response chart. This looks like this. So it gives them the procedure, the materials that they need, warnings because of course you're working with some different chemicals such as ammonia, iodine, a bicarbonate. You don't want them to be ingesting any of that, so you want to make sure that you definitely go over with them what the warnings and cautions are. Then of course you have your procedure. Very simple because you're already going to have your test tube set up and labeled A through F is how I label them. You can label them however you like. Just making it simple, I did A through F for the 6 test tubes. And then you want to make sure they each have a pipette so that they can begin to apply or to mix in the indicator using about 3 milliliters, 2 to 3 milliliters of that indicator solution to their test tubes. Okay, so here in front of me I have my test tube rack and I have my test tubes all labeled A through F. They are going to be then filled with the different liquids that the students are going to have to figure out what solution is inside of that test tube, what liquid is in that test tube. I give myself a small key so that I remember and I have it on hand to make sure that they've got it right as I go around and check their results. But again, you don't want them to know going into it, so don't let them know what it is that they're making. It is fun though, if you are going to create as you create the bicarbonate solution or bicarbonate soda, you can actually do that with the students, and that just requires for them to understand how to make that solution. You can also have them work with the iodine solution. I would recommend just doing the iodine solution yourself, but the students can learn how to do the bicarbonate, and that one is pretty easy to do. So all it requires, you need a glass. You want to fill this glass with warm water. You can kind of see the steam on mine. You don't want it to be boiling or too hot. You just want it to be warm. So this is a little bit warmer than room temperature, because then that way your bicarbonate will actually be able to, or I'm sorry, your baking soda will actually dissolve. Next, you want to measure out 2 tablespoons of baking soda and add that into your warm water. Oh, see if I can get an actual scoop here. There we go. 1 and 2. Okay, and then after that, you will mix that around until it's all dissolved. And something to know is it's okay if your liquid is cloudy. You can actually add more baking soda if you'd like for a challenge for your students. They can actually see the difference in if it's really cloudy as opposed to if it is clear. You can hear it fizzing, getting all dissolved in there. Make sure it's dissolved before you go ahead and put it into your test tubes, because if it's clear, then it helps them because it's looking like, you know, your ammonia or your tap water that you already have in here. So it's almost like they have several different clear ones, like, I wonder what that reaction is going to be. It's definitely a mystery when it's clear. So as you can see, mine is pretty much dissolved. Awesome. So there you have it. Nice and dissolved and ready to put into your test tube. Okay, so the next solution that you need to create is your iodine solution. The only thing to remember with the iodine solution is that you want to have a 1-to-1 concentration. So that means if, for example, you do 20 ounces of iodine, you want to do 20 ounces of water, which it can be from the tap or distilled. It's up to you. So for my purposes today, since I only have a test tube this big to fill, I'm not going to be making a huge amount. I'm just going to make sure that I stick to that 1-to-1 concentration to make just enough for our experiment so you can see it here today. Okay, so for mine, what I'm going to use is I'm going to do 1 Dixie cup and 1 Dixie cup. There's my water. Iodine provided in your kit. And Dixie cup. Okay. Then, of course, want to make sure it's mixed. And the idea with this iodine solution is you really don't want it to be too light, but if you would like to dilute it even more, for example, you could just add some more water and you'll get a different reaction. So that's up to you how you'd like to do that. But again, for your purposes today and what you're seeing, this one, we're just going to stick with the 1-to-1 concentration. All right, so now we've got all of our different liquids in the test tubes and ready to go. All we need is in a Dixie cup or a small plastic cup, because those are also sometimes, I know they were included in my kit. You may have them in yours, those small clear plastic cups, like little plastic cocktail cups. Those are also great to use for the indicator too. Just need a cupful so that the students can, and of course you'll notice when you make your indicator, at least every time I've made my indicator, it produces quite a bit of liquid. And so, with that being said, you can use as much as they need, depending on how many times or how much they need per test tube. So, now, in our test tubes, I'm going to go ahead and grab some of our universal indicator. So then I have that. Okay, universal indicators here. Using the pipette, like I said, you want about 2 to 3 milliliters of liquid, and then they are added to the test tubes, like so. And you'll notice you're coming up with quite a few fun colors. Let me see if I can get my white jacket here to help you see the colors better. And you'll notice the more that they add to them, the more color reaction they will get. So if you do have that much universal indicator, which like I said, it comes out with a lot, then they can put even more into each of their test tubes to really get a strong color reaction, which is, again, always fun. It's always fun. They don't know what to expect. And as they're adding this universal indicator into it, the students really just get an absolute kick out of all these different colors that end up turning up, especially when 4 of these were clear liquids, all looking like water. The only ones that didn't were your lemon juice, would have looked a little bit yellowish. Obviously the iodine, and that's it. So yeah, those 2 were the only 2 that weren't clear. So again, students really don't know what to expect, and you get some really great color changes. Your students can decide to do these one at a time. You can have them do it one at a time and then record immediately their reaction as they go. So putting in the universal indicator to A, recording what the reaction is, B, what's the reaction, C, and just fill the entire part about reactions in first. As you can see here, yep, filling in the reactions. Then, after they've done the reactions, you can bring their attention to the solution chart, and the color reaction chart, I should say. And then that way they can fill in what they think each of the solutions are on the bottom part of that chart as well for their data. Now, if you look, I have the flashlight just to kind of show you a little bit clearer the colors. You're going to have your first one is lemon juice, and that turns out like a bright pink, it says. However, it's going to look a little bit cloudy. You're going to get the bicarbonate will be like an aquamarine or a bluish color. The iodine doesn't seem to really change too much, but it does give a distinct, as far as what they call it, it's a yellowish orange, dark yellowish orange color, which is true for what I'm seeing here. Obviously the water is just going to be a lavender color. And then you have your dark green is ammonia. The vinegar turns a rose pink. So the students will be definitely able to determine the difference between A and B in their colors of pink. Mine actually for the lemon juice turned out a little bit more like a red color rather than a hot pink. But so those are, hopefully you can see them pretty well, the different colors there that you will get with your color reaction, universal indicators, and unknown solutions. Now, the unknown solutions lab, the mystery solutions lab, is very fun to do, but if you don't have access to certain things such as a test tube rack, you can do things a little bit differently. And also, of course, the way that you split up your students is up to you. You can have it, again, be an individual experiment that they're doing, or you can have them work in teams or in pairs. It's up to you how you would like to go about splitting the students up, as well as setting up your materials. So if you don't have access to the test tube rack, then another idea that is given here for the unknown solutions lab is that you could put them all into one cup, and the students have to go around and gather their own samples. So they can be in a cup or on a rack, and the students have to go and get one of each. And just make it so that they get up, they move around a little bit, as well as enjoy figuring out what they are in the end. Because, again, the colors are always the fun part. This leaves room for a lot of expansion. You can go through different discussions. What happened when this, was added? Why do you think that happened? Why might this be something useful for scientists in the CSI field? Because, again, you always want to make sure that they're collaborating and thinking a little bit further as to why is it important, how does it make it so that the information gathered is accurate and objective. So after the mystery solutions, then you go into another mystery lab, which is mystery powders. Mystery powders, you're doing it about the same way. So you will be, here the students will be exploring chemical and physical properties, as well as the changes and reactions in those, as well as they have to, again, make observations, just like what they were doing with the unknown solutions. They have to collect, analyze, and record data. Again, same as what they did during the first mystery lab. And then they seek and weigh evidence to solve problems and draw inferences using scientific principles. All of this is, of course, outlined in your binder. So the Mystery Powders lab is a little bit different than your Mystery Solutions lab. In the Mystery Solutions, you're using the universal indicator in order to see the changes in liquids and solutions. In this, what the students are doing is even further exploring the properties of what it is that they are testing and trying to figure out, of course, what it is. And that being said, what I want to say about that is they are going to do a few different things here. They are not only going to observe, of course, we talked about observation, so they're observing what color is the powder, what does it look like if it's on a piece of black paper, under a magnifying glass, etc. Then you're also going to have them touching it. So they're going to the whole time wear their gloves and their goggles. Their goggles and gloves are very important, especially if you're going to do the expansion on this, because the expansion requires flame, and doing a chemical test is always something you want to wear goggles and gloves for. Then what they do is they pinch a little bit at a time. They feel how it feels like dry. Then they add, for example, the first time they add, 3 drops of water to their Petri dish to see what it is that it feels like after water has been added to it. Each time you want to have them go rinse out their dish and do it again with the next powder. So the first thing is to observe them, test it with the water, and then wash and repeat. Then you get to the chemical property test. The chemical property test, they need to do this 1 powder at a time. The other one, because it's with water, they may be able to do 2 of them at a time if it's just a pinch of that powder. But in this case, you want to make sure they only do it 1 at a time. So iodine for the first powder, wash out the Petri dish, dry the Petri dish, and then do the vinegar test. So there's 2 different ways to test the chemical properties. So the first is to use the iodine, and the second is to use your vinegar, both of which are included in your kit. And you just used, of course, for your Mystery Solutions lab. That being said, we go to each of these powders, put a little bit, just a pinch, into your Petri dish, add the iodine, and see what happens. Just 3 drops, exactly like how they were doing with the water. So 3 drops of vinegar, 3 drops of iodine, 3 drops of water each time that they're testing this particular property. And it's always just a pinch of that powder. After that, they record, of course, always recording. So when they did the water, when they did it dry, they have to make sure that they are very carefully recording what it is that they are observing. And this is something that's extremely important. Again, going back to the whole idea that the data has to be objective and accurate. So that being in their minds that it's got to be accurate, they really need to be careful with their observations. And you can even touch back to when they did the observations of the doctor's office and the murder in the office, and just remind them, this is what we need to do. We need to always be observing. Every little detail matters. It has to be accurate. It has to be objective. And the only way to get there is if you're very carefully recording everything you observe. The flame test itself is totally up to the teacher's discretion. If you are comfortable with doing it, then you may do that expansion. But it's also if you have time. So time, comfort to do this one. It's an optional part to the mystery powders, but it is explained for you how to do the flame test with a Bunsen burner if you have those available. But again, also if you're comfortable, if you don't feel comfortable with the group that you have, depending on their ages, I would not recommend doing the flame test. So then you get to the really fun part of the mystery powders lab. Mystery powders, the students themselves have gone through and they have made sure that they figured out what each one was and what it felt like when they did those 3 different tests, the water and the 2 chemical property tests. They have all this information that they recorded in their chart, which is provided in your binder and in your kit. And now they get the sheet that says Mystery Powders Lab. They have to come to you for 1 baggie of mystery powder. So with your sandwich baggies, again, included in your kit, you are going to go ahead and put a mystery powder in those baggies. Now, they can be all different. Make sure if you're doing that you label the corner of your baggies so you know what, for example, what B is or what A is, if that's the type of coding you're going to use, or labeling, I should say. So make sure you know what it is yourself. Give yourself a key, much like what you did with the mystery solutions, and then hand that baggie to the student to take back to their Petri dish. Then they're going to go through the tests again to try to figure out what it is that you handed to them and really truly discover what was that mystery powder in the bag that they were given. So at the end of your mystery powders lab, the students are being asked to really think a little bit further. You can even go as far as to have them look under a microscope if you have that available. Again, you can always just have them look with magnifying glasses if that's all you have at the time. It's not too big of a deal which way. It's just good for them to take a real close look in some way at these powders. So then, of course, they're going to write down how it is that they figured out what that mystery powder was. So utilizing the information that they had recorded as they were first learning the different properties of the powders in their data table. So they can then hopefully be able to explain in a paragraph and identify the powder correctly. So after you've done all these, now you are into your case studies. Okay, so the first case study is Who Killed Dr. Cosmo? And this is the first case study. You find it in your binder for inside of your kit, page number 19. What's really helpful is that since everything is on a Google document, you are able to print all of the materials that you do need from there. So with that, I printed out and utilized my manila folders. The manila folders just help you to organize everything out because there are several things that I would strongly recommend printing and laminating if you have that availability and that possibility. So here is what it looks like in my crime scene files. So this one is specifically the first case. So there are 6 different cases that can be solved, or 6 different case numbers that each have a different person who is the murderer, that each have different times of death and dates. They have different hours for the time of death, different DNA markers, hair evidence, fingerprints, all of which are different for each of those 6 cases. So particularly, like I said, for this today, I'm showing you case number 1 of Who Killed Dr. Cosmo. Inside of their folder, the students then would find the crime scene photos that they need to observe and collect information from those observations to put towards, of course, the next part of this, which is at the end they're going to have an explanation of who they think did it, why, what evidence, etc. They also have to identify different pieces of evidence, for example, hair follicle and the hair evidence, the DNA. They have to be able to connect that to suspect cases or suspect files as well. So first thing that you will find inside of your case studies are your photos. So there's a photo of the full crime scene. You see poor dead Dr. Cosmo. And those are the full scene photos. Then you have each of the different evidence pieces or evidence markers. So you have 1 and 2, 3 and 4, 5 and 6. 5 is the hair, as well as 6 is, it looks like the drink itself. And you have some shoe prints, which you can choose to use for marker 7. You can choose to use the number 13 evidence of the shoe print itself. And something a little fun was an F- paper, so some different things that were found on the desk there. Really a fun little case. Then it is important, I like to do give the blood type determination chart, as well as to give them the chemical identification results. So this would be so that they can decide what it was that was in this evidence, which is, let's see, reaction, not blood, bright pink color, positive for blood. So they get a discolored stain as marker number 12. Marker number 11 goes with this. 5 and 6 go with these. So you have a connection to each of the different pieces of evidence that are inside of this case. Then, of course, the student team crime response. So they have to then fill in all the information that they've gotten. They have to answer the questions that they've been given to talk about what it is that they've discovered through their investigation of this case. Now, each of them also comes with the different suspect files or profiles. So each of them will have a fingerprint, hair follicle or hair fiber. It'll tell you a blood type, shoe size, everything you need to know about these suspects. So again, there's a lot of information that the students need to observe and collaborate on. So what I like to do during this is to prepare them for that collaboration factor. I like to make sure that they are taking it step by step. One person you work on blood, you work on hair fiber, you work on the shoe print, you work on so I like for them to split up the tasks within as if they are a crime scene task force themselves. That way they are doing things individually, but then coming back together to put everything that they've done together and collaborate on what the result is and who they find is guilty and who it is that killed Dr. Cosmo. So again, helping them to really get that feel of what it's like to be on a crime scene investigators team, and also to investigate as well as come up with answers and discuss the answers to make sure that everyone is in agreement and accurate on the data that they are providing. In a nutshell, there you go. I know it's a lot of information, but the kids really do love these. And like I said, there are several different ones to do, 6 of them, and you can just go ahead and choose whichever one you like. You can switch it up. If they figure out one, you can maybe give them the evidence for another one. So if you do number 1, maybe then you do number 4, etc. Depends on what you feel and how much time you have and how quickly your students are coming up with the answers. Now, they may come up with these answers quickly, but make sure that they are accurately writing down and recording all data and information that they have come across or discovered or collaborated on. They have to be accurate in their writing. Because again, that is something that is reiterated over and over again, said that you need to make sure you're accurate and objective and really have no doubt about what it is you're saying. The end of your CSI experience, Forensics Academy, is the case study, the Who Killed Dr. Cosmo, as well as you do have an additional case study of the murder at the Getty Place Hotel. I like to start with the murder of the Getty Place Hotel, or at the Getty Place Hotel, just to get them thinking about doing case studies first and kind of do it all together and collaborate as a whole group. And then I give, I split up into 2 teams and do the Dr. Cosmo case study. Now, some extensions and my own personal additions to this. I have found that with this entire academy, everything is very, well processed. It's all very organized and in specific order. But there's always room for extension. So I find that when I get to the end, after the case study has already been done, I still have a couple of extra sessions left. So in that case, I like to go over some blood basics with the students. Blood spatter and different patterns and being able to know what type of blood it is that we're seeing. So for that, I have in my own personal PowerPoint presentation that I was showing you earlier, I added some slides called Blood Basics, and I like to discuss spatter patterns with them as just a fun extension. So here's what I have for Blood Basics. So the first thing we do is we talk about what makes up our blood. Red blood cells, white blood cells, plasma, and platelets. And we discuss the different things that these cells and they do within your blood. Then I go into blood facts, just talking about the blood, as well as the genetics of blood types. So now we're discussing blood types particularly. And you'll notice that in the case study with Who Killed Dr. Cosmo, you actually do have blood types that are listed for each of those suspect profiles, as well as for Dr. Cosmo. Okay, so then what are blood types? We talk about that. I talk about the different ones that they are that you could have. And one year I was actually very fortunate to have a phlebotomist come in and discuss it as well. Another year we were able to connect to somebody in the police department. An actual detective came in who has been to crime scenes and talked about those. So there are a lot of things that you can reach out and see what is available to you to make these experiences in each of the weeks even more exciting and more exploratory. Now, how common is your blood type? A lot of the students don't know their blood type. If they do, then this is always a fun thing to kind of show them. Let's see. Of course, talking about blood transfusions and how one person can receive or give blood to another, and then the RH factors. This is an interesting thing to always show them because then it's just a little, again, an extension for them to learn about as well as to apply. Now, talking about blood evidence, and again, that's part of your Dr. Cosmo case study, is there's blood evidence, or just there is blood evidence in the actual murder scene itself, and then there's the blood profiles in the suspects' profiles. So here's how they determine, or the different types of blood evidence that there are. So you have blood samples, which obviously can be matched DNA and blood type, blood droplets to give clues of a location, blood spatter, which we talk about, which the different patterns would determine maybe how that person's murder may have occurred. Then we have a lot of fun with the blood spatter. So if you do have access or you do have the funds available, I recommend that you purchase, especially around Halloween time, at the end of Halloween, if they have, a good sale on it, some fake blood. Because what you can do with the fake blood, and what I did as an extension, having the extra time and an extra week to do it, we actually had the students creating blood spatter patterns. And they were using syringes and dripping, syringes and throwing, etc., to try to create different spatter patterns. And of course, I did show some things, some different patterns, just very basic, like what it might look like if it came from an artery or so forth. Just something a little bit, yes, gruesome, but something really intriguing and interesting to them. And they had an absolute blast creating that spatter and then determining what kind of spatter it might have been. So definitely something fun and exciting. Again, I just came up with that on my own as something to go off of what is so well explained and done in this academy and binder. Of course, you can always go back to the end of your binder, which you do have a fire forensics lab in there. Very, very thorough, very, very well guided lab and evidence and everything that you can do with that. So really a great Explore Labs fire forensics lab for you if you'd like to do that extension and talk about how there is a CSI of fires in general, that there is a person who goes to explore and examine and observe and report on the different ways that fire has been started or spread, etc. So a really great lab, like I said, at the end of your binder after your appendix. Also, you can go back to your CSI experience at any time. That is always fun. And again, it has something for every discipline of forensic science. Just like when it was outlined in the beginning, when I used my own personal one, that CSI, the rice.edu, it's such a fun site, and they really get to explore so much within the different disciplines of this field. Also, there's lots of YouTube extras. If you want to take a look back at each of the different weeks, there's always YouTube extras and extensions that they provided within the binder. I also like to take a look and find things that connect with it on YouTube, so you can always find something that goes along with it. That being said, this is really all I have. That is, in a nutshell, how to teach the Forensic Science and Crime Scene Experience Academy. I hope you had fun, enjoyed my little demonstration, and I hope you have a lot of fun teaching this to your students. Again, it's my favorite, and I've been doing it for about 3 years now, and I really hope that you love it as much as I do. I know your students will be very, very interested and excited to learn and feel like true CSI techs. Have a great day. Take care.